Thank you, Johar, and good morning, everyone. I'm Dinesh Parmar, as Johar explained, and I'm head of regulatory from Polypeptide Ambernath. It's a pleasure to be here at Kyoto, Japan. My talk is basically about impurities in drug substance and the challenges and way forward during product development and commercial phase, considering impurity in the center. So when we talk about a product development or particularly peptide product development, the pendulum of discussion always oscillates towards the impurities, and majority of time having we have different type of discussions. So having impurity specification, control strategy, threshold limits for longer chain peptide, shorter chain peptide, generics versus NCE is always important. And hence this topic, and it's very near to my heart, so let's have a discussion further on this. So disclaimer, the slide shared here represents my personal perspective because I'm going to share some insights which are not yet published or listed in the guideline. The main emphasis on the presentation revolves around my experience with the generics peptide, though a comparable approach can be utilized for the new chemical entities. The presentation is largely centered on the knowledge and experience associated with the synthetic peptides. So agenda would be, we will discuss about the peptide guideline and the variability, impurity thresholds for peptide, the challenges during peptide product development and way forward, and a case study on commercial phase challenge. So if you see the guideline, if you Google the guideline on the peptide drug substance, you will come across this guideline. These are not new. You will have FDA guideline on synthetic versus rDNA origin, which was published in October 2017 and was official from May 21. You have EMA draft guideline of October 23. You have EAP General Chapter USP General Chapter 1503, which describes CQ critical quality attributes and characterization requirement. But what I am trying to bring here is all these are regional guideline, and ICH harmonized approach is not available. Q3A is not applicable for peptide. So traditionally, FDA and EMA follow 0.5% acceptance criteria for unspecified impurity. Though there are monographs available for peptide drug substances, but FDA tends towards the tighter control. So let's jump to the impurity threshold. So if you see, there are various guidelines available, but ICH harmonized approach is not there. And if you take, example of FDA guideline on generics, rDNA versus synthetic peptide, it talks about identification threshold of 0.10%, and impurity above 0.10% should be identified and specified on the specification. And if that impurity is non-RLD impurity, means the safety study is not performed, then has to be go for a safety study with a risk of immunogenicity. If you take on other hand, the EP guideline or EMA guideline, they are okay with identification threshold of 0.5% and qualification threshold of 1%, which means that impurity above 0.5% should be identified and impurity above 1% should be qualified. So you will see that there are a lot of differences in the expectation, and which forms a difficulty for process developer. If we jump to NCE requirements, as I mentioned, historically FDA and EMA consider 0.5% standard for unspecified impurity. So typically identification threshold was accepted to 0.5% so far, but however recently we have seen the cases for longer chain peptide as well as for the shorter chain peptide where FDA is asking 0.10% specification. And mainly, this shift of paradigm looks to be because of the two reasons. One is safety aspect and second is batch-to-batch consistency. One might argue that the safety studies are already performed for NCE molecules, then why 0.1% is necessary? But to my knowledge, it looks like, if you see the level of complexity of the peptide drug substances, it involves 3 layers of reaction. One is activation of alpha carbonyl group followed by deprotection of FMOC group, and then formation of the peptide bond. So this three layer of reaction involves, as the length increases, the number of impurity formation could be increasing, and that is the reason, FDA wants to know, if any impurity which is generated new during the commercial phase or after commercial, during the life cycle of the product, that has to be studied. And that could be studied based on, if we have a specification listed with unspecified impurity 0.10%, so you will have, a monitoring of a specification above 0.1%, and you will know that, if any new impurity is coming. Let us understand this with an example. During the safety or preclinical study, if we studied 7 or 8 impurities of specified, and there were 2 unspecified impurity, 0.3% and 0.35%. But no one can guarantee, of course we can talk about the process robustness, but no one can guarantee about the 100% of process robustness. So during the life cycle or during commercialization, you might encounter with a new impurity which is around 0.4%, and if you have 0.5% specification, that impurity was never studied during the preclinical study. Therefore, and that particular impurity can bring risk of immunogenicity, and that is where I believe FDA wants us to be on more tighter side, so that we will we will identify and specify those impurities, and we can show the batch to batch consistency. If we take CDE China requirement, they are accepting EP standards, so identification threshold of 0.5% is acceptable. I've seen that there are a lot of questions, and they are emphasizing on chiral impurity, high molecular weight impurity, extractable and leachable. So that's from the CDE, and the PMDA is also published one draft guideline last year, and they are also emphasizing to have a ICH Q3A approach, identification and qualification threshold of 0.15%, which is stringent than EP. Yeah. So the main topic of focus, as you see, with respect to peptide drug substance, these are the most important characteristic or attributes we typically study, which includes characterization, higher order structure, secondary tertiary structure, oligomeric state, aggregation state, impurities, and immunogenicity bioassay. So I'm going to focus more on the impurities, and as I mentioned, during the solid phase peptide synthesis, there are 3 layers of reaction, and because of that, the number of impurity can form, if you take histidine, arginine, those are more prone to racemization, so inversion of configuration can take place during the activation of alpha carbonyl group. If you take, the FMOC deprotection, at that time, insertion or deletion impurity can happen. Insertion can happen from your starting material, and if FMOC group is not protected with piperidine, deprotected with piperidine, then you may get des impurity, truncation, oxidation. These are all impurity during the process. Aggregation is a very good topic, and because of high molecular weight impurity, FDA, EMA all are more concerned about this because this can trigger immune response. And my colleague Jar is going to talk about aggregation in detail this afternoon. And there are extractable and leachable impurities which are of concern. So let's take the product development in 2 phases. One is strategic challenges and the practical challenges. Because when we talk about a peptide product development, we typically form a strategy with our colleague with process development and analytical development. And they always ask, what is the gold standard? Can we go for the EMA requirement? Can we develop a global standard where you develop a product which will meet the expectation of US, Europe, Japan, Korea, China, everything, or we need to go for the market specific development? And as I mentioned initially, since the ICH harmonized approach is lacking for peptide drug substance at this moment, it forms very difficult for us to decide whether in which direction we should go. If we are embarking on a journey to develop a product with the 0.5% specification considering EMA requirement, and it's a long process, right? It will take several years, and after some time, your business development team says that you need to go for the US standard. In that case, you will again have to do the rework. And rework means, there will be a lot of things which need to be reworked with respect to the control strategy, impurity synthesis, identify the new impurities, validation, and all those things. So as I mentioned, there is different standards available, and lack of harmonized approach. Company may have to rework at later stage, and changing the regulatory paradigm, because as I see, the continuously new guidelines are coming in, so, and it's going to be a long process development, so we need to be very focused on the change of paradigm. And the evolving knowledge of regulator is also important, because regulator sees that, okay, there is one okay. So evolving knowledge of regulators is also important. You'll see that they provide comment from one applicant to another applicant, which forms a difficult situation. So if you see the way forward on this, of course the situation will vary product to product and company to company. But ideally, I would suggest that we should define a product development considering future needs. So future looks like to be considering the safety aspect and batch-to-batch consistency. So it should be around 0.10%. And consult your health authority, because we know that we can have a meeting with FDA at different stages. So that will be ideal. And your internal regulatory team also should bring intelligence that what challenges and future expectations are coming from the agency. As I mentioned, not only for generics but for NDAs nowadays, regulator have started demanding unspecified impurity of 0.10%. So at least I would say, if we are already embarked on the journey with 0.5% specification, we need to identify what is expected to reach 0.10%. And that would be a savior for the future. An ambition to have a global standard for drug substance. If you have time, resources, cost, everything, then it would be ideal to have a global standard for drug substance to meet maximum market requirement. This can avoid duplication of work. So this slide is about the practical challenges which we typically face during product development. So you will see that as length of the peptide increases, the higher number of impurities would be there. And we have seen practically that, and I will show in the example also, it's difficult to separate the isomeric impurity from the main peak. The coalition of impurity with each other and with the main peak is common, because you might have a good orthogonal method which is separating impurity from the main peak. But you will get cluster before main peak and after main peak somewhere in RRT 0.97, 0.98, and after main peak somewhere RRT 1.05, 1.07. So that's quite common for peptides. And quantitation of a specific impurity amongst that cluster is difficult. If you have some impurity of, say, an example, OXTRP, and your customer would need, that impurity for the study to study in that drug product. So it will be difficult to identify the specific content or quantify the specific content from the cluster. Development of right orthogonal method needs effort and time, and that is important and need of our. With all this complexity, the claiming the right purity and total impurity is always, debatable and challenging. The selectivity, again, if you take the manufacturing part, the selectivity on the purification column to separate this impurity is key to achieve the right yield. And achieving the process economics, considering all these attributes, the tighter controls would be very challenging. So this is one case study where I'm trying to display that this was the EP method on left side. And this particular in EP method, if you will see, the 6 impurities were co-eluted when we performed the peak purity using HRMS. We came to know that all the 6 impurities were under the mean peak. And with the orthogonal method, we have separated the impurities, but you will see that, the first impurity in green, so this was co-eluted with Des-Gley impurity, and because of the co-elution, there was a double count. So we need to subtract that impurity. So we have separated the 6 impurity using orthogonal method with a sign of column, and 6 impurities were comprising of 0.5 to 0.6% even with the EP method, which has huge impact on purity and total impurity. So if you take the purity calculation, we have to typically subtract, if we are claiming the purity with the EP method, we need to subtract all these 6 impurities from the mean peak. And to avoid the duplication of reporting, the impurity was around 0.27%, the green impurity which I highlighted in the previous slide at RRT 0.89. So this was subtracted with this particular impurity, blue impurity, to have right quantification. Let us take a more complex example with 30 plus amino acids, and you will see that between 0.1 to 0.5%, there are almost 19 impurities, and most of them are chiral impurities. You will see that all this impurity were under mean peak with the primary method, and this was separated with method 2, which is orthogonal method. And there were 2 impurity, gamma glue and dipro. These 2 impurities were co-eluted in both the method, so double count was possible. So we need to take care of all these issues, right quantification of total impurity, individual impurity quantification, as well as the purity quantification. So these are the batch data compared with RLD. So you will see that these 2 impurities which were co-eluting were separated from. So basically you have all these impurities at 0.98 RRT and all these isomeric impurities at 1.06 RRT. And these 2 impurities were separated, but these are co-eluting. So gamma glue is there in the first blue, and dipro is there in this co-elution. So we can subtract this level so as to have a right quantification. Can be deducted from the cluster impurity to have right quantification. And isomeric impurity, if you see, if we have not developed the right orthogonal method, this level of impurity was also there in RLD. If you see our batches, which was less, but in RLD it was high. So having orthogonal method is very important. A co-eluting impurity with tighter control, but individual impurity content is difficult, because that's the real challenge at this moment. I'm presenting here, the different analytical tool which we use at Polypeptide from the analytical side. So, having right impurity identification, position determination is crucial to synthesize a particular impurity. And after synthesis, we develop a right method, and then we perform the validation. So it is essential for the specification formation and validation study. This activity takes time and suggested to perform early stage and sophisticated core tools described below. So HRMS is typically, as you all know, this tool is identifying the mass differences, and we can judge the impurity structure. Quantify the impurity contribution in the co-elution impurity HRMS will help. And useful tool if there are multiple impurity under the mean peak, but with the different masses can be identified. So identification and quantification is possible. MS sequencing we are typically using to identify the position of impurity, and based on that we synthesize the right impurity, because there could be several serine isomer, there could be several different isomers. Of course, for isomeric impurity this will not help, but for different impurities this will help. Replacement impurity with the same mass can be identified. As I mentioned, limitation of isomeric impurity with the mass technique. So for isomeric impurity, typically the primary method is chiral GCMS, where you identify the individual isomer. But this is very controversial method, because when you do a peptide bond breaking with 6-normal HCl, racemization or inversion of configuration is possible, and you might get isomer because of analytical error. But at least a good technique to identify the initial what are the potential isomers formed once you manufacture the pilot batches or exhibit batches, and then based on that you can synthesize the impurity, and then again inject in your primary or secondary method which was developed, and then you can perform the validation. 2D LCMS is also a tool where a binary system is available with different column and mobile phase, and helps in the separation of impurities. So basically you will see that the learning and the best practices from all this discussion is effective development of process and analytical method is key. Right first time could be a key and the savior, which can avoid a lot of work. Thorough investigation of impurity profile at development stage can avoid delays at exhibit and filing stage. So if we are developing product at development stage with the right orthogonal method and right purification, then this can avoid challenges at exhibit stage when we are dealing with customer, and when we are actually in the filing stage. You can use all this sophisticated tool as described here: HRMS, 2D LCMS, MS sequencing, chiral GCMS as primary tool to identify the impurity and identify structure and position. Synthesizing isomeric impurity is key, because this is something which we have seen taking a lot of time during development, and if you're not synthesizing right isomeric impurity, you will not have right orthogonal method, and you don't know whether the isomeric impurity is lying under the main peak or is separated. So the investigation should be thorough during development stage. And one may adopt the subtraction approach, as I indicated, for the right quantification of impurity. And for generics, RLD data and impurity identification should be made available well in advance to avoid the complication at exhibit and registration phases. So all in all, if you see, there's more work, more effort, no shortcut, but it's interesting and evolving. So that's all about our discussion on product development. This is a slide on commercial phase case study considering impurity and center. During commercial phase also, you might encounter with a new impurity, as I mentioned, because of complex drug substance and a lot of reaction with the length of the peptide. You might encounter with the impurity at particular RRT where you're already claiming some impurity. So this was during the deficiency phase. We got deficiency from FDA, might be from they might have this particular impurity from different supplier. But when we performed the HRMS study for that impurity, we found that this impurity was co-eluting with one of the impurity which we already claiming as specified impurity. So this was reported above 0.10% for generics. So this is basically I'm talking about generics where we claim unspecified impurity less than 0.10%. So the impurity was around 0.13%, and we found that impurity was non-RLD and non-USP. So the content was difficult to justify. And if you see the situation analysis, there were a number of products, there were a number of customers were already approved in US and Europe. There were customers which were under review phase, and there were customers which were about to file ANDA. So the situation was quite complex considering all the phases of the commercial phase, under review phase, and prior to the filing phase. So the challenges was, assess the impact on the overall API quality, maintain the supply to customer in the commercial phase, because we were already supplying, and with this impurity and with this profile, we had to, stop and pause at one moment and try to resolve this matter. And then notify yet to file customer and, ensure the operation continuity. That was the challenge. So let's see the way forward. So as a polypeptide, we typically follow approach of, whenever such issue come, quality and compliance should be kept on the first priority. And then we try to ensure the supplier reliability to patient and customer, because once we resolve the quality issue, the supplier reliability is important. And then we think about the business and profitability. So in this particular case, if you see, we identified the root cause. So the process analytical process control strategy and analytical control strategy, both are important. So based on the process, we identified this was the DES impurity, and we took, the proper control to, increase the FMOC protection, by which we proved that this impurity will not be there. And then analytically, we developed a right orthogonal method to separate this particular impurity from the co-eluting impurity, so that we will ensure that all further batches will not have this impurity. And surprisingly, when we checked this particular, retrospectively, the batches lying in the market and batches which were in the stock, we found that, impurity was found in only one batch. So there was not a lot of issue. Impurity control at in-process stage, and we avoided, API specification change. So this was the main savior in our case. No impact and change of document at customer end. This avoided major variation and impact at customer end, and notification to customer was proposed to early stage and post implementation. So outcome was change was approved change was approved by health authorities, and ensured that impurity is controlled in the batches below 0.07%. Therefore, no impact at customer end. The change executed and implemented without any business impact. So if you see the keynotes during life cycle of the product, such issues may arise, but addressing them in a compliant manner is crucial, which required right strategy and approach. And not revising the API specification in this case was savior. Learning and preventive measures should be leveraged to other products to avoid such issues. So typically, I like to highlight that regulatory intelligence, regulatory strategy, proactiveness can keep company and customer ahead of time. So lastly, I would like to, this presentation was not possible without the support of my team in regulatory affairs, Peter Lawson, Abhishek, Patil Pawar, Manisha Beluse, from PD and AD, Jon Rasmussen, Linda, John Billing, project development, Charlotte, Ankita, Ambernath QC, AD, Abharat Ravindra, Anket, and Ambernath production, Atul Harshe and Harshal Devdar. With this, thank you so much. Thanks for the patience and listening me.
Thank you, Dinesh.